Right. This I didn't know about your talk, and it's actually quite relevant. And Gary called his model 2.0, and I'll take a more humble position. I call my model beta. All right. So I will start with that. Thank you, my fascinating collaborator, David Brown, that we worked together for this project. We're both from Michigan. And I am interested in modeling the visual cortex and trying to align the models with the visual brain, and especially when the model and the brain deal with naturalistic and complex stimuli. In particular, stimuli that we are working with, there are large scale and movie stimuli, and about over a decade ago that we assembled back then a very large video data sets that has about 13 hours of videos, and we recorded from 3 subjects and done this for many months. And one of the subjects was myself, and one was assistant professor. I spent a lot of time in the MR scanners myself. The first type of analysis we're doing is to see that when human brains process the same movies as the convolutional neural nets, and how do they compare with one another? Can we actually use those models as a decoding device to help us understand what it means when the brain activity is showing different patterns? The first thing to do is that we, when the model processes the same movies as the humans, and we use the model's activation to predict the brain responses. In this case, it's the fMRI responses. We use linear projection models and regularized with sparsity constraints, L1 norm. And it turns out it works really well. And back then I was really surprised, and I couldn't believe that this works so well. So even though at different locations, they process very different visual features, and they show very different temporal dynamics, and they can all be predicted pretty well. And you can see that a large portion of the visual cortex, or even beyond, can be well predicted. This includes both the ventral and the dorsal stream, and that was another surprise because the model is a nice model trained for object recognition, and it is not a good model for the dorsal stream to begin with. It captures many of the functional organizations, including the retinopathic organization and the mapping of the different levels of visual processing. It also maps the face processing networks, and without doing those face localizers task paradigms. And this could also, to some extent, decode the brain fMRI responses and reconstructing some visual patterns and categorical representations. So it captures both the semantics and the visual features. Some improvement over the years, and there are these days one of the popular topics that see papers in Neuroscience and iClear reporting increasingly better performance on the visual decoding. That's not the main purpose of it, and I come back to a similar set of points that the brain there's really a lot of different gaps between the models and the brain, and a few that we trying to fill or address is that first, the retina is not a simple camera, and there are at least two parallel pathways. So it's not a single model, unlike a convolutional neural net or vision transformer, and then we often learn without being supervised. So this is a foveated vision. This is why very relevant to Gary's talk that happened just now. So we tend to have a lot of visual acuity only to the very center where we look, and their peripheral regions operate in a very different way. So this is a log polar sampling and transformation. The retina definitely have a lot of dense samplings around the fovea, at least for the parvocellular pathways. The magnocellular pathways has a more broad and wide views. If you take those log polar samples and transform back to the original input space, you see that our brain actually sees the whole world through the fish eye. So you can see a lot of really walking. And so the black areas is because, once you walk onto the retinal sampling and walk with the back, and you're going to have a lot of empty space on the very the side of it, so we cannot have pixel values assigned to those locations. So why is this relevant? This makes the where to look important decision that we make that every day, right? Even though we do not feel so, so that we walk into a new scene and where we look, there must be a strategy that the brain has developed, and we want to figure out what that strategy is. So part of the strategy is that the wide view will have to kind of figure out where to look next. And that's based on the information of the aware system in the visual cortex, as a dorsal pathway. And this is largely segregated from the Watt pathway, which go along the ventral stream. Putting these two things together, and there are two largely parallel pathways, and at least there should be two different neural networks, and how do they work together is a question that we want to figure out. So we built a model. The model has two convolutional neural nets. One we call the aware CNN, the other is called what we call the Watt CNN. The aware CNN and start with the type of retinal samples that has a wide view. We're trying to mimic the magnocellular retinal ganglion cells. You take a lot of wide views, and we figure out that the where should be the next location that eyes should be fixated to. And then that information pass to the Watt pathway, because then the Watt CNN will need to look at around a small narrow view and around the fixation, and then zoom in to that local areas and then sample the visual information. So in some way that the where CNN just keep visiting different parts of the visual scene, and the Watt CNN trying to draw visual input and build the representation cumulatively through, in this case, a GRU, a few layers of a GRU that accumulate where the visual information across different eye movements. So this one shows again that the where and guides where to look next. Now, how do we train this model? We train this model for object recognition. It's still supervised learning. So how do we do that so that there is where to look become what we want to optimize? The way we trying to train the fixation is such that we want the model, the two networks combined together, trying to make a correct perceptual decision as fast as possible. Okay, so we use the reinforcement learning strategies to train this strategy. So the where CNN has to learn to capture, it has to learn, the good fixation points, so that the Watt CNN will draw useful informations and combine those information across as fewer eye movements as possible to build a scene understanding. All right, and then the model was trained using ImageNet and MS-CoCo. One's object as is the scenes. And after we trained, this is what model does. So this is where the models figure out the visual salience and where are locations that eye movements tend to focus on. And then we show this. This is a movie stimuli, Indiana Jones, and we play this movie to the in the fMRI scanner, and we record fMRI responses. And we also compare that where the model look versus where the human's eye movements compared to human salience and attention. And this seems to match very well. In the bottom row, it shows that where the model look in a sequence. We'll start from the red and go to yellow and then blue. So for each scene, there's a few possible eye movements. And then once we build a model, we'll see how this model compared to the brain. So the strategy we use is to have an encoding model. So we look at where CNN explains, what CNN explains for each voxel, and then we look at their contrast. This is what the result shows, and it turns out that the where CNN tends to explain a set of regions in the brain that largely around the dorsal visual pathways, and the CRCNS tend to explain better the voxels along the ventral pathways. And then different area, different ROIs seem to fall under this kind of binary case, so that in the midline is that an area that is equally well explained by the what versus the where CNN. So you can see that there's a consistent set of areas that explained by one model versus the other comparative model. And we asked the question that what actually explained to this, because the model have two sets of differences. One that they serve different goals, that's the learning objective. One trying to figure out location, the other trying to figure out perception, the object identity. The other difference is that they take different inputs, right? So one is the broad view and the magnocellular retinal ganglion cells. The other is the paulocellular retinal ganglion cells. So we did this ablation studies. It turns out that there are different functions are the main drivers why this one model better explain the dorsal versus the ventral ROIs. Okay, that's what we do. And the other thing that I want to touch on is that that we humans apparently for most of the time that we learn without supervision. We don't need people to tell us or teach us where to look, right? So how does that happen? And some of our inspirations are going back to how Newton figured out the gravitation by looking at an apple falling to the ground. So what is that inspiration? So if you look at, if you think about a series of events, right, like a ball or an apple falling to the ground, you see the position of the apple is y, right? And if you do you look at the y changing its position, so that's why y is a function of t, and you do the Taylor series extension of that, and you'll figure out that at any points you have a position which is y, you have a velocity which is v, and you also have the acceleration which is the g. So this is and you can continue on, right? You can, but then the fact that Newton figured out there's a gravitation is that somehow in his brain that he drew this inference regarding the fundamental causes of the position, and it's a first order derivative, second order derivative, third order derivative, and so on and so forth. And we think of the different derivatives is a different view of the world, all right? And then if we take all this, different views into a model, and a model need to draw a conclusion. That conclusion is about a fundamental cause that drives all the different views. And there's only one representation. In this case, that representation should be indicative of the gravitation. So that's where our inspiration is. So then we did this, for the image recognition. So we have different videos, and we look at how the video, in this case, I call this videos as X. We look at the first order derivative over time and look at the derivative at different time frames as the second view, and the original video frames is the first view, and the second order derivative as the third view. And then you can do this, in many different views, but in this, in our initial exploration, we stopped at the second order, and we train a model. The model needs to collapse different type of inputs into a consistent representation, invariant of the views. So in this case, the Taylor series expansion is the data augmentation we use. And we train a model using a self-supervised learning loss. That's the instance discrimination. So the model need to figure out that this set of views are coming from the same videos versus, and then so different videos, and no matter what kind of views, they need to actually map onto different locations in the representational space. When we do this one, and we realize this model learns much faster with very small, requiring very little data that we can learn, and relative to some other, self-supervised video learning, like the contrastive learning and the BIC regularization, different comparative models. This seems to be a very efficient ways for learning abstract representation. It also makes sense that when we look at where the model turned to look at in the input space, that some of the models that they can look at, for example, the backgrounds and some of the areas that are shortcuts in order for the model to do well in the instant discrimination. But this learning strategy will allow the model tend to look at really important positions that make sense to our visual behavior. Last but not least, that we're not just doing fMRI, we are also doing, when I say we, it's actually David is also doing the intracranial EEG recordings from epilepsy patients with implanted electrodes. And by far, we have recorded 54 different IEEG data set from 54 different patients. And this includes this video movie stimuli and among others. One of the hidden opportunity is that the videos are played not silenced, it has audio and a visual. So David and his team have shown preliminary findings showing that they can use the IEEG to decode the phonemes, and the decodability would improve by 10% when the speakers is shown in a visual cue in the movie stimuli. So there's stay tuned. Maybe next year he will come and to CRCNS to present these findings. With that, I'm happy to pick up questions, and also thanks for the CRCNS for funding.